I will today remember to hit record. And, oh, sorry, I forgot to let all of you record, right? Kind of distracted. I'll just go through the list. Jessica, Joe, Kira, Melissa, shouldn't have a recording, and then Raj. Okay, I think I got everyone. All right, so apologies for that. Okay, make sure that's recording this time. All right, so now these are what usually textbooks call extensions of Mendelian genetics for a single gene, and that's what your textbook calls it as well. Many patterns of inheritance are not as simple as what we have already discussed. So the first extension that I would like to talk about is one called degrees of dominance. So far, all of the problems that we've looked at have what we would refer to as complete dominance, meaning one allele is completely dominant over the other, right? So if we have this, if we have a heterozygote like that, we will see purple because it's completely dominant over white. Now, that is not the only type of dominance. There is also something that we refer to, so if we label that like A, there is also what we call incomplete dominance. And this is just like you would imagine. It means that the dominant allele cannot completely mask the recessive allele. And what we mean by that is that we would see a blended or a new phenotype instead of just purple or white. And the example that your textbook gives is usually snapdragons. And we would take, the example is always a true breeding red flower crossed with a true breeding white flower. And the F1 generation, so we would have this. The F1 generation would all be heterozygous. And if this was a completely dominant trait, we would expect them to all be red. But in this case, they are all pink. So they show an intermediate phenotype in this case. Sometimes it's the shape of the leaf, like a broad leaf and a narrow leaf, and the in-between is a medium. And usually when it's incomplete dominance, the in-between is some other version of a color or a characteristic. You will see in your textbook that they change the symbols. They do it this way. They do C for color, and then they superscript capital R like this. And then they actually switch the allele to white for the recessive. I find that to be a little confusing at this stage in Bio 1. This is more something that we would spend more time on if we were in a genetics class. If you want to do that as your notation, that's fine. Like I said, I find this simpler, and I can clearly understand either one is fine if you want to use it when you work on the problems. The reason we say that this is incomplete dominance and it still follows Mendel's laws is because in the F2 generation, I'm going to need a new whiteboard here. In the F2 generation, it's not blended inheritance because in the F2 generation, our white flowers come back. So in the F2 generation, we would have the three phenotypes of one that is red, two that are pink and one that is white. So this one would be RR, this one would be pink, and this one would be white. Like that. So that's one of the difference of degrees of dominance, right? So I mentioned that we had complete, incomplete, and then the third would be what we call codominance. And that's when both of the alleles are dominant and expressed in the phenotype. So neither one is recessive, okay? So there are two examples here. The first one is a protein that's expressed on the surface of blood cells, of red blood cells, and the protein is labeled M or N. And notice I've drawn them as both capitals. So an individual, if an individual was heterozygous, they would look like this rather than a capital and a lowercase, right? And then, of course, all of their offspring would have, they would either be all M, maybe we got two that are M, N, so expressing both of those proteins, and one that was only expressing N. Codominance also occurs in blood types in humans. In the case of blood types in humans, there are actually 3 alleles though. So it's an example of not only, it's an example of 2 things, codominance and multiple alleles. So let's get a new board for that. So we have 3 alleles for blood type, A, B, and O. And again, the notation changes. When you see it in your textbook, it's going to be depicted like this. The letter, capital letter I with a superscript of A, capital letter I with a superscript of B to indicate they're codominant, and O is going to be indicated by 2 little i's because that is the recessive trait. So we have 3 alleles, 2 show codominance. That would be the A and the B allele. And then there is 1 recessive allele. Each individual, each one of us, we can only have 2 of the 3 alleles, right? Because we're 2N, not 3N. So we're going to get some combination of blood type A, or A carrying the recessive O. Right, so this would be like, in your book, you're going to see it looking like this. Or same for blood type B. We might get blood type B that we have 2 copies of the same allele for B. We might have BO, which would be depicted like this. And it could be AB, because again, they are codominant. Or you can be blood type O. So this one would look like this, capital A, capital B. And then blood type O would look like this, 2 little i's. Again, each person, we can only be one of these, right? And then depending on who we mate with, that will influence what the possibility of alleles are for our children, right? So let's do a quick example here. If if there was a woman who was blood type A, for example, and she married or mated with someone that was a man that was blood type B, and how could it be possible that they have a child with blood type O? Right? So the only way we can get a child with blood type O, you kind of have to work a little bit backwards, right? You have to put O here, and you have to say, okay, how can I get a child with blood type O only if each of these parents is carrying the recessive allele, right? So they would have a 1/4 chance of getting a child with blood type O, and they'd actually have a 1/4 chance of getting a child with any of the possible blood types. That's a B. Some of you might be aware there are other alleles, there are other proteins and carbohydrates that are expressed on the surface of red blood cells, and one of them we call the Rh factor. That's a separate characteristic, right? So if we included that in our discussions, then that would be a dihybrid cross. And we would need, of course, all 16 boxes for that one. Okay, so we have complete dominance, incomplete dominance, codominance, and multiple alleles. And there's one more type of problem that I'm going to ask that you learn how to solve. It's called a sex-linked trait. I'm going to put codominance here. I'm going to erase this and put multiple alleles. And then the last type that I wanted to talk about real quick was sex-linked traits. So this is chapter 15 and plus a bunch of other things, but the only part of chapter 15 that we're going to focus on is the sex-linked trait. And that means, just like it sounds, it means that the trait, the characteristic, what we were calling character, right, is on a sex chromosome. So in humans and in a lot of other animals, right, sex is determined by a pair of sex chromosomes, and XX determines a female and XY determines a male, right? So because the X chromosome is so much larger than the Y chromosome, it actually has more genes on it. Most sex-linked traits, most, not all, most, are physically located on the X chromosome. So what does that mean? Well, what that means is that females are often what we refer to as a carrier, meaning they carry a copy of the sex-linked trait, but they have one normal copy of the gene, so it's hidden, right? Females are often carriers. Males, we see a higher proportion of males expressing the characteristic. This work was first done in fruit flies, and the trait that was studied was eye color. And the normal or wild type eye color was red, and the mutant eye color is white. So male flies were more frequently white-eyed than the female flies. And I'm not going to go through that whole example with you. I would rather do maybe a characteristic in humans that's a little bit simpler to understand. Sorry, Joe, I didn't click on the right button here. Chat. The first second, it doesn't matter. You can put them wherever you want. I usually just do it that way because it's easy for me to remember, but you can put them, doesn't matter if you flip them because you're going to get the same answer anyway. So let's look at color blindness in humans, because that's a sex-linked trait. That's an easy sex-linked trait. And it's more complex than this. There are many different degrees of colorblindness, and now we know there are more genes involved than a single gene, but I'm just going to pretend that it's simple. So those traits, so we might put a plus to indicate that this is a female, a normal female, like that. And then I usually put a circle around the X when we do these problems so that it does not confuse with the X's for the sex traits. So let's say we have a normal female, a normal vision female, let's say, and she mates with a colorblind male. So we're going to give him the C gene for colorblind. And let's say they have, what are their chances of having a colorblind boy or a colorblind girl? And that's all going to depend on what this female has for her second allele that she's carrying, right? So let's say we have a normal vision female marrying a colorblind male, and let's say their first child is a colorblind boy. Okay, so if we were drawing our Punnett square, right, and then the question might be like, what are the chances they're going to have a colorblind girl, or what are the chances they're going to have another colorblind boy? And now we would separate them by the sex characteristics. So we would have the male here, and put him on the top, and the female down the side. And we said she had normal vision, right? But they have a colorblind boy. So fathers can't pass their X chromosome onto their sons. They can only pass their Y chromosome onto their sons. Boys get their X chromosome from their mothers. So she must be carrying colorblind trait in order for them to get a colorblind boy. Oops. Like that. So here's the colorblind boy. And then I could ask you any of the questions, right? What are their chances they'll have a colorblind girl? 1/4. What are their chances they'll have a normal visioned boy? 1/4. What are their chances they'll have a normal visioned girl? 1/4. Great. So this would be an example. When it's a sex-linked trait in the problem, it'll usually be identified. It'll say blah, 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 blah is a sex-linked trait. And that's how you know that you're going to have to set up your Punnett square and use the sex chromosomes specifically. Okay. That's the last of the different types of problems that I would ask you to solve. There are some more vocabulary words. I'm going to leave that. I should leave that. There are some more vocabulary words that I wanted to go over with you. And those words are pleiotropy, and epistasis, and polygenic. Okay, so I'd like to give you definitions for each of those. You'll find them in your book. They're words in bold, of course, right? So pleiotropy is when you have one or a single gene, but it has multiple effects on the phenotype. And I think your book gives an example for the character determines flower color actually also affects the color of the coating on the outer surface of the seed, which can be gray or white. So that gene for flower color is also determining something else in the phenotype. Epistasis, this is when one gene actually controls the expression of another gene. And in this case, it's usually the same part of the phenotype. I don't know how else to say that. The example they give in your book is coat color in Labrador Retrievers. And there are 2 genes that control coat color. I'm just going to write in dogs because that's easier. And it turns out that they are the B gene and the E gene. And if you take 2 heterozygous parents, so big B, big E, little e, and you cross them, in the F1 generation, we would normally expect 9 to 3 to 3 to 1, right? We would expect 4 phenotypes, and it turns out this would be black and brown and yellow, maybe, and like completely albino. But it turns out that you end up with 9 to 3 to 4. And that's because one of these genes, the E gene, controls whether or not the B gene gets deposited in the hair of the animal. So we end up with 9 black dogs, 3 chocolate dogs, so 3 brown dogs, and 4 yellow ones. And that's because when you have when you have 2 little e's together, when 2 little e's get joined together, that prevents the color gene from depositing either black or brown in the hairs on the dog. So anytime there's 2 little e's, this gene actually controls whether or not the B gene gets to put down the pigment. So all the dogs that have 2 little e's, they are all yellow. And this is drawn out for you in figure 14.12 in your book. And you can look at it and read the example. I'm not going to ask you to solve a problem with epistasis. All I expect you to understand is to recognize this proportion and be able to recognize the definition of epistasis. And polygenic is just like it sounds. That means many genes influence, sort of the opposite of pleiotropy, influence one character in the phenotype. Sorry. Like height, for example, that's not controlled by a single gene, it's controlled by many. All right, now to trace a gene in a human or in maybe in a dog or a cat where you don't have a lot of offspring in each generation, geneticists employ what we call a pedigree. And a pedigree is a chart or a way to, like it's like a flow diagram in order to follow a particular trait through multiple generations. And there are a couple examples in your book. I think they give you someone with a widow's peak and someone's whether they can taste PTC on their tongues. It's kind of a bitter taste. And for pedigrees, what you need to know is how you make the diagrams. So square is males, circles are females. And if the person is affected, we say, so if they show the trait or characteristic, they're colored in like this. Most books use either purple or red. If they're a carrier for a trait, meaning they're heterozygous, then only half of them is colored in like this. Okay. And then matings are depicted by a line. between the two individuals. And then children of that cross or that family, they are depicted like this in birth order. Okay, so here's two people that have 4 kids. Birth order is boy, girl, boy, girl. Right? If there are twins, depends on what kind of twins they are. If they're twins that are from the same zygote, goes like this. If they're not, then they would just be in birth order. Who was born first? And then again, they would be colored in or not colored in, and then we could follow a characteristic or trait perhaps through multiple generations. So they have all girls, right? So you'll see some examples of that. And I don't know if I included a problem like that in the homework. If I didn't, I'll try to make one up and put it in, because I know on the quiz that's a possibility that you'll have to have a pedigree or do part of a pedigree to figure out a particular trait. And that's where we're going to end chapter 14. So that'll be all finished. And that's the end of what will be covered on the quiz on Tuesday. Tomorrow, like I said, I thought we could have some time to do genetics problems together. Unless people have questions already today, I'd be happy to go over some of those today as well. So I'm going to go back and continue the recording then. So I would ask you some sort of a blood type question, most likely. So keep in mind all of these. Usually the question is, a woman with blood type A marries a man with blood type B, and they have a blood type O child, and the husband thinks that she cheated on him. How can you help this couple find out that it's possible that they may have given birth to a blood type O child? Or a woman who is blood type A marries or mates with a man that's also blood type A, and they have a blood type O child. How does this happen? Or a person who is blood type O mates with someone who's blood type B, can they possibly have an AB child? Right? All of those are the questions. This is the Punnett square that you'd have to figure out the genotypes of the parents were, make the Punnett square, right? And then figure out, just then be able to answer the question. Yes, you should know A, B, and O, absolutely. And like I said, this is the more formal way of them to be written. And yeah, I would know that. Absolutely. That's pretty much the only multiple allele problem that there is in Bio 1. We talk about other ones in genetics, mostly a lot of them in plants. But for us, that's the only, if it's a blood type problem or some version of a blood type problem, that's what I'm asking. You're welcome. Anyone else? All right, then, guys, I'll see you tomorrow if you want to go over some problems. If not, have a good weekend. I'll see you Monday. I'll try to get caught up on all the grading so that you know where you stand. And that's it. See you Monday or tomorrow.